Good evening, this is Mr. Wintermelon. So we are on this chapter of biological molecules, and what you have learned in the previous video is the role or the function of water in living organism, why is it important, and how it plays a part in animals as well as plants. In this video, we will be focusing on carbohydrate, fats as well as protein. Why I am showing you this document on the syllabus outcome is because I want you to take note of what you are expected to learn and the focus. So the focus here is you need to know the elements which make up these nutrients or biological molecules. Then next is the test for the following nutrients: starch, reducing sugar, protein and fats. The describing part, in this case, I would say I don't think it's very important that you know how to write down what you are doing. Never seen it in the written paper. Practical, they just introduce the year-end practical, so not very sure, but it's definitely not part of SPA if you are concerned. For the carrying out part, I would advise you to just practice in a lab, because watching video is not as good as hands-on practice. Then you need to know which molecules, which large molecules is made up of which small molecules. So you need to know the basic unit of the macromolecules. So let us start with the first nutrients, which is carbohydrate. Before we start, my advice to you is to copy down this table, because it will help to summarize your learning, as it will cover the important aspect of the syllabus. So you have your carbohydrate, fats as well as protein. As you have seen in the syllabus learning objective, you need to know the elements of carbohydrate, fats as well as protein, the single unit that make up carbohydrate, fats as well as protein, then the large molecules that make up the three nutrients as well as the food test. So please copy down this table into your notebook before we begin with carbohydrates. Carbohydrates are organic molecules that are made up of carbon, hydrogen and oxygen. So you will see the three most abundant elements inside our body appearing a lot today. It is useful but not essential for your O-level syllabus to know that carbohydrate follow this chemical formula. So what it's trying to say here is the number of hydrogen is twice the number of oxygen. So for example, you have 10 oxygen, that means you have 20 hydrogen in that carbohydrate. That's about it. So for carbohydrate, they are simple sugar as well as complex sugar. Simple sugar are those carbohydrates that are made up of one or two basic units, while the complex sugar are those that are made up of long chain of glucose. For simple sugar that are made up of one unit, we call them the monosaccharide. Two basic unit is known as the disaccharide. Mono means one, di means two. So these two are the simple sugar, while the complex sugar, we call them polysaccharide. Poly means many. So there are three monosaccharides that you have to know. The most important one is glucose, followed by galactose as well as fructose. Disaccharide, there's maltose, lactose as well as sucrose. So you can see the molecular diagram. Monosaccharide, there's one unit of sugar, while disaccharide, there's two units of sugar. If you ask me, the important sugar to take note is glucose as well as maltose. These two are the most important out of these six simple sugar, followed by sucrose, which is the regular sugar, the common sugar that you use for cooking. So how are the disaccharide made? Maltose is made up of two glucose molecules. Lactose is made up of one glucose and one galactose, while sucrose is made up of one glucose as well as one fructose. Other things to take note is lactose, normally you only find it in animal, while sucrose, you normally find it in plant. As for polysaccharide, we have our starch, cellulose, which you have learned make up our cell wall, as well as glycogen. So the new word here is glycogen. What is glycogen? For plant, they store their food, their glucose, which they produce during photosynthesis, as starch. But for human and animal, we do not store glucose as starch, we store them as glycogen instead. So these three polysaccharides are all made up of glucose. Long chain of glucose molecules. So please copy all of this down into your table that you have written. For the single unit, you write the glucose, galactose, and fructose. For the secondary unit, you write the maltose, lactose, as well as sucrose. Then for the large molecules or the macromolecules, write in starch, cellulose, as well as glycogen. Now, let us move on to the tests that you can perform on carbohydrate. First is the test that you should have learned in your elementary or primary school, which is the iodine test. It is used to test for starch. As for the result, just take note, when there's a positive result, all is good, you will write, oh, iodine turned blue-black. When there is no result, please do not write there is no observable change. Write instead, starch is absent, iodine remains yellowish brown. Do not write there is no observable change. And blue-black looks like that. However, please take note, in O-level, this type of practical question did come up before. In this case, instead of using iodine, they use very diluted iodine solution added to starch. It will still turn blue, but I don't know so much of black. It will definitely turn blue. It will not be yellowish brown. Then they say we add amylase solution to it. You do not need to know amylase solution right now. You will learn it later. But what is going to happen is the amylase is going to break up the starch, so you will not have starch anymore. So when starch is absent, iodine is supposed to turn yellowish brown. But in this case, when very diluted iodine solution is used, normally you will not observe the yellowish brown. Instead, you will become a clear solution because the iodine is just too diluted for the yellowish brown to be observed. So just take note of this. While we learn that by default, if it's a theory paper, if iodine is absent, it's yellowish brown. But when using very diluted iodine, because of the dilution, the yellowish brown might not be clear at all. So don't be too clever and write yellowish brown when you can see that it is actually colorless. So just take note of this. The second test for carbohydrate is the Benedict test, and it is used to test for the presence of reducing sugar. Reducing sugar is just a fancy way of saying simple sugar minus sucrose. So it consists of 5 simple sugar: glucose, galactose, fructose, maltose, and lactose. And the easy way of remembering this is GGFML. G for glucose, G for galactose, F for fructose, M for maltose, and L for lactose. Sucrose is a non-reducing sugar. Why is it non-reducing sugar? Or what's the meaning of reducing? You will learn that later on in chemistry. I will not go through what's the meaning of reduce in this video. Not necessary for your biology. And the test is more complicated than iodine test. What you have to do is to add water to the food sample. Then hopefully the sugar will diffuse out. Then you add in Benedict solution and put the whole mixture into a boiling water bath for 5 minutes. It is very important that you remove the mixture after 5 minutes, because if you put it for too long, the result might be different from the actual result. And if you just leave it in a water bath that is being heated by Bunsen burner, the sugar will actually decompose and you will not get the red precipitate if the reducing sugar is present. Instead, it will become black because the sugar decomposes as you have leave it in a water bath that is being heated. If reducing sugar is absent, the solution will remain blue. But it's important down here to take note that it's not red coloration, it's break precipitate. That means there are some solid stuff going on. And the thing about Benedict test is actually there's a range of results. So here you can see the method. We have a solution of, let's say, reducing sugar. Then we add in Benedict solution, put it on a water bath. The diagram is not very good, and actually this one is from your textbook. Please do not put the mixture on top of a flame because the sugar will just decompose. And just leave it there for 5 minutes. Then, if there's no reducing sugar, it will still be a solution that is blue. When there's very little or trace of reducing sugar, it will be green, precipitate. If there's low level, it will be yellow. If there are more, it will be orange, precipitate. And lastly, if it's high, it is red precipitate. So this is for carbohydrate. Let's move on to fats. Lipids, or more popularly known as fats, are stored in our body. It can provide us with energy like carbohydrate as well as keep us warm. Similar to carbohydrate, they are made up of the following elements: carbon, hydrogen, and oxygen. So the basic unit of fats is glycerol as well as fatty acid. Write this down inside your table. And molecules of lipids will look like this. The glycerol is the 3 carbon on the left, while the fatty acid are the 3 chains that you see down there. Each chain is 1 fatty acid, and each corner represents a carbon atoms. For the test of fat is called ethanol emulsion. What you have to do is to chop up the food sample, then you add in alcohol or ethanol into the food sample, shake it vigorously. What it's doing down here is to allow the fats, if any, to dissolve in the alcohol. Then pour the alcohol out without the food sample. Then you add in water before shaking again. If fat is present, then you should get a cloudy emulsion. If fat is absent, the mixture should remain colorless. For this ethanol emulsion test, to be honest, in practical, I haven't really seen it coming out before. For your written paper, it is probably going to be MCQ if it comes out. So at the end of the day, you need to know the result and what it represents. That means if they say, oh, food test, there's cloudy white emulsion, what does it mean? It means fat is present. That's about it. Then lastly, we have our protein. So the basic unit of protein are called amino acid. So amino acid will link up to form polypeptides, similar to how glucose link up to form its large molecules. Polypeptides is considered large molecules already. So write down in your table, polypeptides under the macro or large molecules. Then polypeptides will fall into the form of protein. So basically polypeptides and protein are the same thing, just that polypeptide is not folded while protein is folded. For amino acids, they are made up of the following elements: carbon, hydrogen, oxygen, and nitrogen. That's why nitrogen is the least out of the 4 most common elements that are found in our body. And sometimes there is sulfur in amino acid. The sulfur part, not very important, but I'm just showing it out to you so that you can kind of know. For your monosaccharide, there are 3 monosaccharides. For amino acid, there are a lot more. In fact, 20 or even more. Their basic structure is like that. So you have your standard amino group as well as your standard acid group. The only things that change between different amino acid is the side chain, or what we call the R group. So the R group can be very varied. As you can see, the R group can be non-polar, polar, as well as acidic or basic. Basic means alkaline. So this different amino acid will give each unique protein its own property. Do you have to know this? No, you don't have to know. You just need to know that different protein will have different property. And this is mainly attributed to the different type of amino acid that make up the protein. As for the test of protein, it is called the burette test. What we have to do is have a sample solution, then we add in the burette solution. Just shake it, very easy. If protein is present, a purple or violet solution is formed. If there's no protein, the solution will remain blue. And again, I like to emphasize, if there's no result, do not just write no change, write what you see, which is the solution remains blue. And this is all for this chapter. I may add in some lab practical if I get a chance. If not, we will move on to next chapter. And if you have noticed in the syllabus objective, it will be on the thing called enzyme.